anything recorded in this. Oh, it wasn't recording yet. Oh, good. That's good, because that's the kind of thing that would end up like, as evidence. Your Honor, I was kidding. Famously good defense that, gets people exonerated all the time. Okay. Electron transport and chemiosmosis. Where have we left off? The reason I'm not finishing yesterday's note is because we're kind of just going to pick up where we left off. Now, what has happened so far in the process of breaking down sugars? When we are using here the example of metabolizing glucose. Your body has pathways for metabolizing all sorts of nutrients, but it is all the same by the time we get into the mitochondria. So which step might be different? We used glucose as an example. What's the pathway for glucose? Glycolysis. Glycolysis ends with what two molecules? Pyruvate. What did it start with? Glucose. Hey, that's a question on the quiz. You got that. In the process of glycolysis, we take glucose, which is one of many sugars we can ingest. And of course, sugars are one of many food sources we can ingest, right? We can also eat protein. And do we get energy from proteins? Yeah. So that means proteins we're going to have to get into the mitochondrial, into the mitochondrion as well. So it doesn't go through glycosis exactly, but it will end up doing everything that came after that. Fructose is a food we can eat. Fructose actually enters glycosis. It just enters already at stage 2. Because what's the first thing that happens in glycolysis? Well, first we phosphorylate the glucose, and then we use an isomerase to change it into fructose, which is an aldose instead of a ketose, which means it makes a more symmetrical ring. We do that because when we cut it in half, do we immediately get two perfectly identical molecules? No, but they're pretty close. They're both how many carbons big? 3. And then the first thing we do once we've cut them in half is we use an isomerase again to make sure that they now are identical. The DHAP, the dihydroxyacetone phosphorylase, gets turned into that G3P like the other one, right? And then from there on, they go through the same cycle to end up as 2 pyruvate molecules. Where does all that happen? Cytoplasm. Good. Hey, that's another question on your quiz. Look at that. Look at that. Didn't study, didn't get ready. Nailed it. Nailed it? Nailed it. Feels good, right? It feels good when you get a question right and you knew it. It feels a little better when you get a question right and you just totally guessed, right? If we're being honest, if we're telling the truth, we can admit it, right? Feels like you've cheated the system. Anyway, yeah. What do we call that? What's the process by which it goes into the mitochondria? It's a paired reaction. And we're going to talk about that more today. These enzymes are often multi-purpose, which is if you're going to build a machine to move pyruvate into the mitochondrion, you might as well also build it to do another step of its modification along the way. So what happens to pyruvate as it moves into the mitochondrion? The extremely exciting part where I talk, and you don't want to miss it. It's very simple. It's called what? Pyruvate oxidation. Pyruvate oxidation. And we haven't talked about this yet. We're going to talk about it a lot today. But oxidation doesn't necessarily involve oxygen, which makes it one of these stupid words in science. Interestingly, the Latin words in science are always really good. They just describe what things are, but you have to know Latin. The English words are often just like either the name of the guy who did it first, or the first white guy who did it, or they're like the first thing they used to discover it, which often isn't even the thing. So like oxidation doesn't have to involve oxidation. And when we took grade 12 chem, what's an oxidation reaction actually about? Yeah, what about them? In oxidation, what happens? An atom will be oxidized if its electrons get lost, if it loses electrons. What do we call it? The thing that takes them away, the thing that gains them. It is reduced. It's a reduction reaction. Those are always paired up. So in pyruvate oxidation, we steal some electrons from pyruvate. And in the process, that means it's oxidized. Something's going to have to be reduced. And the thing that gets reduced is this NAD molecule, NAD+, which gets reduced to something called NADH. That's one of 3 molecules we use to store energy. So at the same time, the pyruvate is losing electrons, the NADH is gaining them. The NAD is gaining them to form NADH. And in the process, it can attract a couple H plus ions, which are just floating around in the water. If you take grade 12 chem, you'll learn that there's always H plus ions floating around in the water, so you don't have to go make them. They're just always there to be found, just like we assume there's always water molecules to be found, right? When we went through Krebs cycle, we needed water, but we assume there's always water available. So at that point, the pyruvate actually, in the process of losing those electrons, to facilitate losing those electrons, it has to break a bond. What happens to the pyruvate molecule as it's oxidized? It loses electrons, NAD becomes NADH. We just got some more energy from it for the cell to use. That's good. That's why we're doing this in the first place. But the electrons that break that bond, this is one of 3 times we get sort of a special type of bond breaking where we actually lose what? Sorry? Nope. Carbon. This is one of the times we lose. How many carbons in our original sugar? Now we're working with half of that sugar. So how many carbons do we have to deal with? 3, and we just lost one of them. Just in the act of going into the mitochondrion, we lost one of them. What's it get released as? How many carbons do we have left to work with? 2. They're in what form? An acetyl group. A little 2-carbon group called an acetyl group. It's carbon with 3 hydrogens, and the second carbon that has a double bond to oxygen. It's got an aldehyde group on it. But that doesn't, if we just left that by itself coming into the mitochondrion, it would float all over the mitochondrion, but we want to be able to control where it goes. So that enzyme that takes the carbon off and reduces the NAD to NADH also does something with that acetyl group. It does what to it? It sticks it to something. What's it stick it to? Not itself, because that protein's embedded in the membrane, but it sticks it to a different thing called coenzyme A. And coenzyme A is a coenzyme. It's not actually a protein. I misspoke and said it was yesterday. I knew it wasn't. Not all enzymes are proteins. The overwhelming majority are. But we have this class of things we call coenzymes, and they kind of function with enzymes. They don't have to be protein-based. This one isn't. It's sort of like a big steroidal molecule, vaguely phallic-looking, and it sticks to the acetyl group. And now it gets moved into the mitochondrial intermembrane space, where what process happens? We learned about yesterday. Krebs cycle, also known as the citric acid cycle. So called because there's this molecule in the inner membrane space called oxaloacetate. It's how many carbons long? Nope. 4. And it's going to get stuck to that acetyl group, and now it's going to form a molecule that is 6 carbons big. That's called citrate, or citric acid. It means the same thing. Citrate now is going to go through a series of reactions just to get turned back into oxaloacetate. Why do we stick it to a bigger molecule? This is something I never learned when I was in high school, but I think it's useful to actually think about the why of it. Yeah. Yeah, it's just much easier to manipulate a big thing. We do this, right? Like if I'm working on electronics that are really small, I get out a clamp and I clamp it to a structure that makes it bigger and more solid, right? We put things on conveyor belts. A conveyor belt's a really big thing, even though the thing we might be working on is pretty small. It's easier to work with things that are big and stable. An acetyl group is going to float around. And then once we take the next carbon off, whatever you're left with is really going to float around. It's probably going to be a gas and just want to float out, right? So if we stick it to a bigger thing, it's easier to work with and get that energy out. So we stick it to oxaloacetate, forming citric acid. And I'm not going to go through all the steps, but we know then it gets isomerized into isocitrate. And at that point, we're going to use a dehydrogenase. What's a dehydrogenase do? It takes a hydrogen away. Of course, to do that, it has to take electrons away, but that's okay, because where is it going to put them? It's going to take the electrons out of that isocitrate, and it's going to put them into NAD+, forming NADH. That's another oxidation reduction. Our sugar, or what's left of our sugar, is getting oxidized. It's losing electrons, which also is going to mean it's losing energy. And to do that, we break a bond. What gets released? Another carbon dioxide, right? This is another place where we lose a carbon, right? And those electrons go into the NADH, and well, the sugar remnant gets oxidized, its energy gets lost in the form of electrons. The NAD+ molecule gets reduced. We store that energy. Do you think we get all of the energy? 100%? Is it perfectly efficient? What is some bound to be lost to? Entropy. Some of it's bound to be lost just into the universe, but we get most of it. Then we have a second dehydrogenase right away, which is going to take that last carbon off. And now we've dealt with all the carbons from our original sugar. Are we finished immediately then? We actually go through a few more steps where we change the shape of that thing, eventually getting it back to oxaloacetate. And each of those is progressively a slightly lower energy state. It's not actually quite true. 2 of them actually go up a little bit in energy, but I don't have to get into the hyper details of it. During that course, we get the last bits of energy out. Altogether from the citric acid cycle, we get NADH out at 3 points. We get another molecule reduced. Does anyone remember? FADH becoming FADH2, actually. You'll get it memorized eventually, don't worry. That's another one of those oxidation reactions where the remnant of our sugar is oxidized, it loses energy, and the FADH becoming FADH2 is reduced, and that's gaining energy. Again, we're storing energy. And there's one more in there that looks a little bit like ATP, but instead of being based on adenosine, which is a modification of adenine, which is one of our little nitrogenous bases we use in DNA, this one's based on guanine, right? Which is the other of the purines, the other of the single ring nitrogenous bases, and double ring nitrogenous bases. And that adenine, or that guanine goes from being GDP, guanosine diphosphate, to GTP, guanosine triphosphate. And when all is said and done, our sugar is dealt with. Yes, sir?
I just questioned in the class, anybody not complete the DSBN survey, the census survey?
Do you want to go do that now?
Yeah, I'm just collecting anybody who didn't do it. We just need 10 minutes.
Anyone else? This will be recorded, so you can catch up on this. Yeah, if you didn't get your survey done, go with Mr. Matola.
And just come to the cafeteria?
Okay, so today we will deal with capturing the energy that was stored in FADH2 and NADH during glycolysis. How many NADHs did we get from glycolysis? Right? Pyruvate oxidation. How many NADHs did we get? 1 per pyruvate, but of course, how many pyruvates per glucose? 2, so we get 2 NADHs. And then, Krebs cycle. And of course, what's the other name for Krebs cycle? Citric acid cycle. And what did we get there? 1 time through Krebs cycle, how many NADHs did we get? You can go back and look if you don't have it memorized yet. If we go back and look at it, during Krebs cycle, we get NADH when isocitrate is dehydrogenated and CO2 is released. We get it when alpha-ketoglutarate is dehydrogenated and another CO2 is released. And then we get it right at the end when malate gets turned back into oxaloacetate. So we get how many NADHs per cycle? 3. But of course, how many cycles do we get for every glucose? 2. So that's a total of 6 NADHs. And then how many FADH2s did we get during that? We get FADH2 just once when succinate gets dehydrogenated into fumarate. So we get 1 FADH2 per cycle. And of course, how many cycles per glucose? 2, right? Both pyruvates. Well, both acetyl groups, I guess. So 2 FADH2s. Sorry? Nicotinamide adenine dihydroxy, and can't remember. Well, but I'll tell you what it is in your textbook reading. I should know that, so I'm gonna skulk over to the textbook and tell you. Yeah, the old textbook section I gave. Yeah, I totally should remember what it stands for. I have a degree in this, for God's sakes. I used to be able to draw all these, but I'm having a moment, and I can't. So I'm going to swallow my pride and check the notes. Would I be more or less impressive if I acted like I remembered and told you to look it up condescendingly? Should I have just pretended I knew and said, oh, I guess somebody didn't do the reading. Anyway, we'll move on. In fact, can we just go back in time? Well, I guess somebody didn't do the reading. You should probably do that, eh? Anyway. Sorry? the record, it's not like I would expect you to memorize that from the reading, so like that would be an entirely BSE condescending thing to say. But then I think I'm kind of a BSE condescending person, so like I should have been here for it. Anyway, I will look it up and get back to you. But yeah, nicotinamide adenine dehydrogenase is the first, not dehydrogenase, see, dihydroxy. And actually, it's worth looking at the structures of these things too. So we'll go to my friend MolView. God, if you guys had any idea how amazing it is to have MolView, when I was an organic chemistry student and like a biology student, and you wanted to know what one of these things looked like, you had to break out your model kit, which we all had to buy in first year. They probably still make people buy them. And we had to build things. So this is its oxidized form, right? There it is. So that's NADH. And you can see right away, it's a lot bigger than the ATP and GTP complex, right? Yep. These are phosphates. Like the orange itself is the phosphorus, but then those are phosphate groups, which you can see here. And this is an interesting case where there's actually an ester bond between two phosphate groups, right? It's actually a diester bond, a phosphodiester bond, which we see when things are bound at a phosphate group, right, like that. And then if you want its reduced form, NADH, you're going to see that it looks quite similar. In fact, I'll challenge you to go in and spot the difference. There obviously is one. Yeah.
Where does that GTP end up becoming ATP?
We hand wave it in the mitochondrion. There's mechanisms that are able to just, you have little enzymes whose job is to have a GTP become a GDP and use that to energize an ATP becoming an ATP. It's just not very exciting, but it's literally this little enzyme that does that. And I think it has a name like a transferase or something. It's fun. But anyway, so remember, these reactions were NAD plus becoming NADH, right? And the other one was FADH becoming FADH2. These were the reduction reactions where these were energized. And what we want to do today is oxidize these and instead reduce what molecule? Because what molecule serves as the energy source immediately for the cell? ATP. Yeah.
The cycle thing says it's FAD to FADH.
Oh, yes, sorry, it is. I do that sometimes. I'll do it again. Yeah, this one has a 2 on it. The NADH doesn't. Thank you. So what we want to do today is we're going to couple reactions. And a coupled reaction, what we're going to call a coupled redox reaction. Yeah. I'm doing it for my health. I'm doing it so Sam's hands fall off. And what we're going to try to do is we're going to try to have, for example, our NADH be oxidized to NAD+, and at the same time, and I suppose I should mention that that releases an H+ ion, but that's okay, it doesn't really matter, well, kind of matters. And at the same time, we're going to couple that, and this is often how we show a coupled reaction, with ADP and a phosphate group forming into ATP. Now, when we talk about redox reactions, what that means is this. This is being oxidized, and this one is being reduced. And these always happen in pairs, because if you remember, it's all about losing and gaining electrons, which for the purpose of this course, we can think of as losing and gaining energy. Does that make sense? As one of these is giving up energy in the form of electrons, the other one is gaining energy in the form of electrons. There's other ways to give and lose energy, but in this one we're using electrons. And the electron transferring is actually going to eventually allow this bond to happen. Basically, to stick another phosphate group on, we need this adenosine diphosphate to have an extra electron that it can use to form a bond with another phosphate. Does that make sense? So we're going to transfer that electron along. If you need help remembering this, if you've taken grade 12 chemistry, you probably already remember this. Oil rig. oxidation is loss. And in this case, you can think of that as electrons or of energy. Doesn't really matter which, because for the purpose of this course, it's true. And then rig. Reduction is gain. Fun fact about gain, it doesn't have two G's. And again, you can think of it as gaining electrons, or more generally, just as gaining energy. What's the other one we use sometimes? Leo says grr. Leo being a lion, I suppose. I mean, it could just be your friend Leo. My next door neighbors have a kid named Leo. It's not a name you see very often anymore, but he's a cute kid. I say that, he's like 17 now. He was a cute kid. No one at 17 is cute. Present company accepted. Yeah, how's that? And then this must be the place. What is this place? What are we looking at here, of course? Mitochondrion. And we already talked about this briefly yesterday, so I'm not going to go too much detail, but the mitochondrion is double membraned. And this allows two separate, I'm going to say like solution conditions to be maintained, or solution states to be maintained. Huh? What's that mean? Separated by a semi-permeable membrane. Basically, inside the cell, you've got the mitochondrion, and the mitochondrion itself is like two different sacs of fluid, one inside the other. It's almost like your beaker. It's almost like we did that lab meaningfully. It's almost like your beaker where you have a container with solution in it, and then inside of that, you have a membrane that has solution in it. Does that make sense? So you have an inner and an outer membrane, which means you have a fluid between the two. We call that the intermembrane space and the intermembrane fluid. And we have what we call the matrix of the mitochondrion, which is inside the inner membrane. So we have the intermembrane space and we have the matrix. But what's really useful here is that if you imagine this, okay, so here's the outer membrane of the mitochondrion, and then here's the inner membrane. It's folded into these kind of folded shapes that we call cristae. Why would we fold that? What does that increase? The surface area. God bless you, somebody pays attention in math. Why would we want to increase surface area? Where's all the action actually going to happen? On the surface of the membrane. Does that make sense? What we're going to talk about today happens on the membrane surface. So by having this folded like this, we have more membrane surface. So we have the cristae. We'll make a tiny little note and say folds called cristae increase surface area. I've never bothered to look up what the Latin root of cristae is, which is not like me. I always look up Latin roots of things. So I should get on that. So anyway, if we imagine then that we have a solution between the space called the intermembrane space and the intermembrane solution, and then we have the matrix, and we have the solution of the matrix. I actually don't know what the name of that is. I should look that up too. I don't think I ever knew what that is. We always just said into the matrix. Maybe they just call the fluid itself the matrix. I don't know. Could the space and the fluid both be called the matrix? Are you allowed to do that? Well, I was just going to say, I think I just went to university like in 2001, and 1999's The Matrix was still very, very fresh and new, and it was just exciting that there was something in science class actually called The Matrix. I actually took a philosophy class in university, which was the philosophy of the matrix. It actually wasn't a very good philosophy class, but that's neither here nor there. Okay, what I want to focus on is that this right here, if we take just a little snapshot, this is where the business end of this thing happens, all right? Because what you have is a membrane, and it separates the matrix which I'm going to arbitrarily show on the left here because that matches where I drew it in the diagram, and this purple fluid that I'm calling the intermembrane fluid. Everything we're going to learn about today happens with proteins embedded in this inner membrane that do the work. So proteins embedded.
Mr. Winger, please contact the office.
And the whole thing that we're going to drive today is with something called chemiosmosis, or the chemiosmosis hypothesis. What's osmosis, of course? We just did a lab about it. Sorry? Yeah, the movement of water across the semipermeable membrane. And chemi, of course, refers to chemicals of different things. So this is everything we're going to do now. The cell has built a machine that lets us store energy as ATP, and it does it all by moving things across membranes. Which, if you think about it, is a pretty good way for your cells to design a machine because it already has all kinds of mechanisms that know how to build membranes, and it already has all kinds of mechanisms that move things across membranes. Does that make sense? So this is a real recycling of tools. The cell has to have membranes anyway. The cell has to have proteins that move things across membranes and pump things across membranes anyway. And now we're going to use those tools to form ATP that the cell can use to do whatever it wants. Does that make sense? Okay. So let's take a look down at just what I drew here. We're going to look at this inner membrane. And here it is. How are we feeling? Yikes, indeed. All right. And here's what we're just going to make sure we do. This side here is the innermost portion of the mitochondrion, which we call the matrix. Very good. So this side is the matrix of the mitochondrion. And the outer space here is the intermembrane space. Note where we aren't is nothing is happening with the outside of the mitochondrion. Does that make sense? This membrane is not the membrane that separates the cell from the mitochondrion. It's the membrane that separates one side of the mitochondrion from the inside of the mitochondrion. Does that make sense? This would be the equivalent of the little cell bag that you made, which didn't allow the things in the cell to interface with the world around you. It allowed them to interface with the fluid inside the beaker. And the beaker would be the equivalent of the outer membrane of the mitochondrion. Does that make sense? Yeah. So during Krebs cycle, that happens. Yeah. So all of the products of Krebs cycle, because it happens in the matrix of the membrane, are already in the matrix. Now the truth is, there's a couple of things that aren't already. What do we have to actively move into the matrix? Everything that happened during Krebs cycle is already in the matrix, because Krebs cycle happens in the matrix. Yeah. Yeah. Not the ATPs, because those are already ATPs, but the 2 NADHs we produced during glycolysis and the 2 NADHs we produced during pyruvate oxidation have to be moved into the matrix. All of the other products are already in there. So if we go back and look at our accounting, we already have 6 NADHs and 2 FADHs in the matrix. We're going to move in 2 NADHs from glycolysis and 2 NADHs from pyruvate oxidation. Does that make sense? Altogether, what now will be in the matrix? 10 NADHs and 2 FADHs. Does that make sense? Okay. What happens with these? And the answer is redox reactions. So here's what we do. NADH will come here and it will be oxidized. All right? NADH gets oxidized. And again, remind me, what does it mean if we're oxidized? We lose an electron. Where do the electrons go? And the answer is the electrons enter this enzyme. This enzyme called complex 1. All right? Complex 1. Now, at complex 1, these electrons give it what? If NADH is oxidized, that means complex 1 is going to gets reduced. And if it's reduced, it means it's gaining electrons, but it also means it's gaining energy. Does that make sense? And if it's gaining energy, this machine can be used to use some of that energy. Does that make sense? So it gets these electrons, and it's going to give them away. If you look here, it's going to give them to this molecule Q, which is called quinone. But before it gives them to quinone, it's going to use some of the energy. And it's going to use the energy to do what? To pump an H plus molecule, also known as, not an H plus molecule, an H plus ion, which is also just what? Yeah, if you're a physics person, you don't think of that as an H plus ion, you think of it as a proton. It's both. It uses the energy to pump an H plus ion from the matrix into the intermembrane space. Why the heck would it do that? Well, we'll get to that, but that's what it does. By the way, this actually ends up being a bit of a net neutral because, of course, when we turned NADH to NAD+, when we oxidized our NADH, what did we release? H+. So we have a steady supply of H+ ions. Does that make sense? But the electrons get passed on to quinone. Quinone is mobile. If you look at this, this is like that question in the last test I gave you, quite pointedly. This section of it has to be hydrophobic. And of course, these sections of it would have to be hydrophilic. And it's all the way through the membrane, so it doesn't move very well. It can shift around in the membrane, because remember, it's the fluid mosaic model of the membrane. The whole thing is able to kind of shift and move. But this would move relatively slowly. But this quinone molecule, is it hydrophilic or hydrophobic based on where it's located? Hydrophobic, and it can move much easier amongst the lipid tails. And so quinone will actually, it is designed once it has, without electrons, quinone is designed to be highly attracted to complex 1. But when quinone gets electrons, it changes shapes and properties, and it's now repelled by complex 1. But it actually finds itself in a position where it's highly attracted to complex 3, which is a molecule called cytochrome B. We'll refer to it by both terms. Cytochrome B and complex 3.
Would all the students going to the Youth Wellness Hub with Mrs. Mitchell please make their way down?
This also uses energy to pump an H+. And I'm not even going to say it, but which way does it pump it? From the into the intermembrane space. We have now moved to H pluses. By the way, this doesn't release an H plus at this stage, so now we haven't even replaced it. We are starting to move H pluses, and we're not even replacing them here. Does that make sense? And then here, we're going to note two things. At the last step, cytochrome B will pass its electrons on to cytochrome C. And cytochrome C, like quinone, is mobile, though quinone was mobile where in the membrane? Inside of it. It was highly hydrophobic. Cytochrome C is also mobile, but it doesn't sit inside the membrane, it sits on top of it. Is it hydrophobic or hydrophilic? Hydrophilic. And it has the same trick, where it's designed that when it doesn't have those extra electrons, when it's in its oxidized state, it's highly attracted to cytochrome B. But it's designed such that when it receives those electrons, it undergoes a change, and now it's repelled by cytochrome B, but it's highly attracted to complex 4. And so it will bind to complex 4, and it's designed that when that binding happens, what does it do with those electrons? Passes them off. And what does complex 4 do? It also uses energy to pump H plus ions. Now, the result of that is the electrons here got all their energy from NADH. Of course, where did NADH get its energy? Well, depending on where it happened, from different molecules, but all of NADH's energy comes from our original molecule, which was glucose, right? In one way or another, NADH had glucose's energy. When it releases these electrons, it gives their energy, the energy from that glucose, first to complex 1. And complex 1 uses about a third of that energy to pump an H plus ion, where it gets passed to quinone, which will now be attracted to cytochrome B, which is complex 3. And you might say, why does it go from complex 1 to complex 3? Don't worry, we'll get there. And then complex 3 is also an H plus pump. And again, that's why it has to be this transmembrane protein, right? It has to transition across the membrane because it has to bind to an H plus here and shove it out the other side. And then it gives it to cytochrome C, which binds to complex 4. And complex 4 also shoves an H plus 2, but complex 4 does another thing. It also catalyzes another reaction, which is what? Well, the answer is those electrons have to end up somewhere. Does that make sense? You can't just keep passing the electrons forever. And now we've used up most of their energy. So we have to give those electrons to something, and they're not high energy electrons anymore. They're pretty low energy electrons, which means we're not going to be able to make a big high energy molecule. We have to build a pretty low energy molecule. So are we going to build a big molecule or a small one? Small one. What's a real small molecule we could build with stuff that's already floating around in the cell? The answer is we could build water. What do we need to build water? Well, we need an oxygen molecule, right? And then we need hydrogens, right? But we don't have hydrogen gas floating around in the water of the cell, but what do we have? We have H plus ions. If you took grade 12 chemistry, you know that some water always dissolves itself. So you always have H plus ions floating around and you always have OH minus ions floating around. Does that make sense? Well, it just so happens that this is perfect because the only thing that would make this not form water is if these did come together, you would get H2O, but of course those hydrogens were both not hydrogen atoms, they were hydrogen ions. So you'd get H2O, but it would be missing 2 electrons, which by the way isn't stable. But as it happens, NADH doesn't just give 1 electron. I didn't write this earlier. I should have. It actually gives 2. That's just the way it's designed. When it's oxidized, it releases 2 electrons. When it's reduced, it gains 2 electrons. So here, what is it able to do to complete this puzzle? Well, what actually happens in this reaction, and we're going to write it out in a bit more detail, is we're going to say 2 H plus ions, and this is always in the water. All water always has H plus ions. We call it an acid if it has more H plus ions than usual. We call it a base if it has fewer H plus ions than usual, but all water has H plus ions. And we react that with half of an oxygen. Now that might seem ridiculous, but the long story is that this actually waits until it has 4 electrons to work with to make this work, and it uses 4 H pluses. But for the purpose of what we're doing here, where do we get the oxygen? Yeah, this is brought into the cell from the air or water, depending on what type of, but it's brought into the cell from the outside world. We get it by doing what? We breathe. How do fish get that oxygen? They don't breathe, but they run water against their gills, and that water has some dissolved oxygen. They can get it out. How do grasshoppers, do they breathe? No, they have a small enough body that they can just have their bloodstream run close to an empty section that they have between their shell and their inner body, and they can absorb oxygen that way. That works because they're small. You can't scale that up. That's why when you watch a movie and they show like really big insects, you know, like the size of dinosaurs in like science fiction movies, that's not possible because of the square cube law, which is they depend on having a high surface area to volume ratio, so they can just absorb oxygen passively without having to breathe. And once you get past a certain size, that can't work. So insects can only get so big. And at a certain point, their bodies just wouldn't work. And the bigger they get, the less efficient they are. Smaller insects, much more efficient than large insects, because they don't have any mechanisms for actively moving gas. We used to, in grade 11, dissect grasshoppers as well as pigs. And I'm thinking about ordering some this year so we can. It's really delicate work, but it is really cool. And then it needs 2 electrons. And it gets these from complex 4. which came from complex 3, which came from complex 1, which came originally from the NADH, which came originally from our sugars. Does that make sense? These electrons can be traced all the way back to your glucose. So these are from NADH, and really, they're from your glucose. And what does that form as a result? Water. Great. All right. And why does it require energy to move H plus ions? Well, what do we know about H plus ions and our semipermeable membranes? Now, in our semipermeable membranes, how did the ions move? How did our iodine ions move? They moved great. But our membrane wasn't a real membrane. In a real membrane, do ions move well? No, why not? What made our cellulose membrane different from the real phospholipid bilayer?
That thing is made of cellulose. This is not cellulose. This is also.
Yeah, and cellulose is polar all the way through. But this has that feature of being polar, polar, non-polar. And ions cannot make their way through that non-polar middle region, so you need something to move them. But these aren't just passively pumping them. They use energy to pump them, which means they can pump them even if the H plus ions don't want to go there. As this keeps happening, what's going to happen to the ratio of hydrogen on one side to the other? We're going to get more and more H plus in the, what's this space? Intermembrane space, and we're going to get less and less H plus in the, in fact, if you look here, for each NADH, let's track H plus ions. Altogether, what happens? Well, how many do we pump out? So if we assume that we started, this, these numbers aren't real, but let's assume that we start with the intermembrane space and the matrix. Let's assume that we start with 0 and 0, right? Let me try this again. That was a terrible, terrible table, but that's because I'm doing this on the fly. This isn't in my practice note, but I'm just thinking now that I like this idea. Let's do this instead. Let's look at the intermembrane space and let's look at the matrix. At the start, let's just call it 0 and 0, which doesn't mean there's 0 H plus ions, but we're just calling that the default number. The truth is there'd be trillions and trillions, right? Because we're dealing with, molar levels. At complex 1, what happens? Well, at complex 1, 1H plus gets pumped into the, 1H plus gets pumped into the intermembrane space, but 1H plus also gets released by NADH. Do we see that? So here we go to +1 and +1, right? And that's because this pumped one through and this released one. All right, complex 2 doesn't do anything. Complex 3 is another pump. And in complex 3, we pump another one here, right? So what does that bring us to outside? +2 now. And nothing changes here, so that stays at +1, right? Then what happens at complex 4? At complex 4, we have pumped out another one, right? Oh, sorry, I'm silly, because of course if one got pumped out, then what had to happen here? It had to get lost, right? Because we had to move one. So what would this become now? 0, right? And we pumped one out here, which is going to bring this to 3, right? But then what else did we do here? We used up 2 of them. Does that make sense? So we actually lost 2 of them, bringing this down to like -2. So by the time we're done this, there's a net difference, and actually I should have made this 0 because we gain 1 and we lose 1, which means that by the time we're done, there's a net difference of 6. Does that make sense? Because, sorry, if we look here, we gained 1, right? We made 1, but then we also gave it away, which means we lost 1. This would have been smoother if I'd rehearsed it, but again, I'm making this up on the fly. But the numbers work. So there's a net difference of 6. Does that make sense? There's a net difference, when this is done, of 6. So after this process, there is a high concentration of H plus in the intermembrane space. And a low concentration of H plus in the matrix. Is everyone comfortable with me using square brackets to mean concentration? I've done it at least once before, but I want to make sure we're all aware. Should I make a little note here? Reminder. Concentration. But we know from our membrane lab, if we have a high concentration of one thing on one side and low on the other, what wants to happen? When we started that lab, we had a ton of iodine on the outside and none on the inside. What happened? It moved in. Now again, will those H plus ions just be able to move back on their own? Because H plus ions can't move through the membrane. So this creates what we call chemiosmotic pressure. This is a chemiosmotic gradient. I.e., we have high concentrations of something on one side of a membrane and low concentrations on the other, which creates chemiosmotic pressure. That's just a fancy way of saying the H plus is going to tend to try to move back into the matrix. Does that make sense? The H plus is going to be drawn towards the matrix. H plus drawn to matrix, but can't move through the membrane. So, what's that mean? Well, it means that basically what we've done is we've just made a big H plus pump. By the way, the only difference is FADH2 has a little less energy in it, so its electrons wouldn't have enough energy to drive all 3 pumps. And so, complex 1 isn't designed to bind with FADH2. What is designed to bind with FADH2, if you look at this diagram? Complex 2, which leads to quinone, which transfers it to complex 3. So every NADH pumps how many H pluses? Every NADH. 1, 2, 3. FADH, entering the cycle later, will only pump 2. Does that make sense? NADH pumps 3 hydrogens. FADH2 will pump 2 hydrogens. Yeah.
Is that why the amount of ATPs we get from each, say we use 30 and 2?
Yeah. That's right. Now, let me ask you this. The electrons that are making this projector screen work right now. That projector is working. It almost stopped working for a millisecond there a while back. Where are those electrons coming from? The wall. I illegally, I ordered them to install a projector here for a while back, and they were just taking forever, so I snuck up and took the ceiling tiles. There's no plug up there, so I have a big extension cord running through the ceiling, and it connects down in the prep room back there. If we follow that wire, it goes into the wall. If we follow that, it goes into the breaker. If we follow it from the breaker, it goes into the main panel. If we follow it from the main panel, it goes to the transformer on the street. If we follow it from the transformer on the street, it goes to the bigger transformer, sort of in that big central place, in Welland, where there's all the big power lines. If we follow all those big power lines, where am I going to get eventually? I'm going to get to Niagara Falls. How do we get those electrons? We spin a big wheel. What spins the big wheel? Water. It's not actually the falls. What we do is we divert most of the water from the falls into those big hydrocanal rivers. You see, it starts way up here in Welland, and we let that water go, and instead of going down the falls, we have it go down a big tunnel at the bottom of which we have a big turbine that spins. How do we get energy? We get electrons by storing a bunch of water up in one place and letting it fall to another. People are finally catching on to this. Do you know what they're doing in places where they have frequent power outages? When power demand is low, they're making fake lakes, fake reservoirs, and they're just pumping tons of water up into it. And they're covering those with white ping pong balls so it reflects sunlight so less water evaporates. That's pretty clever, right? And then on days when power demand is really high, they're actually building hydroelectric dams. And they're not saving any energy. In fact, they're losing it because we know about entropy, right? They're having to use electricity to pump that water up. But what they're doing is they're pumping it up there when we don't need a ton of electricity during slow times, so that during peak times when we need extra electricity, we can just open up the dam, let some water flow through, and we can get some energy. We're making a battery out of lakes. In the meantime, some places are doing that, not covering it in white ping pong balls, and they're also just letting it also be a beach and stuff when it's not in heavy use and you just cordon off the section by the hydro dam, you know? Kind of a good idea, right? This is all the cell is doing. What it is doing is it's making not a waterfall, but an H+ ion fall. What it's doing is pumping all this H+ into one side, removing it from the other so that those H+ ions are desperate to get through. And they will get through. But when they get through, just like our water falling from a high energy state to a low energy state, when you fight entropy, you get back energy, right? We've created this incredibly low entropy state by moving all these H+ ions to one side. And then when you let them go back where they're going to go, they're going to give you energy. So how do you capture it? And this isn't some funky chemical reaction, not really. It's so much better than that. God, I can't get over how much better than this. It's a physical machine. It is a little thing that spins inside your membranes, and this is completely true. We've proven this by modifying the DNA and having it attached to it, a big whip-like tail, and we've actually been able to film this whip-like tail spinning around, because this thing is a mechanical machine that takes H+ ions going through, uses it to make this thing spin, and that spinning motion attaches a phosphate group which is stuck to it to an ADP which is stuck to it. It's mechanical. Isn't that glorious? Just think about that for a minute. Your cell makes most of its ATP not even through a real chemical reaction, but through a physical mechanism that involves something falling through a hole and making something spin. That is exactly how our power systems work. Now ours use magnetism and electromagnets. This is almost better. It literally uses this thing spinning and changing shape a bit to go like, oh, now you two are stuck together. And an ADP and a phosphate group are now an ATP. And we call this molecule ATP synthetase. So we're going to learn about that real quick. We'll mention quickly FADH2 enters at complex 2. So each FADH2 only pumps 2 H+. So complexes 1, 3, and 4 are the pumps. You might not even need to write this if you can just read the diagram, but not a bad idea. So the electrons that start out in glucose end up in water. And the carbon that starts out in glucose ends up as what does the carbon that started in glucose end up as? Carbon dioxide. Interestingly, the hydrogens in glucose aren't actually necessarily the hydrogens of water that get released, because it happens way down the line. The CO2 that you breathe out is the glucose molecule that was burned. But the water that gets produced by this happens way later. It happens here, and it is functionally impossible that that's the same hydrogen that was in the glucose. You know what I mean? These are just random H plus ions floating around. There's a 0.0000% chance it would ever be the same H plus ion, which is kind of interesting. So when we've always drawn that, like C6H12O6 plus oxygen forms carbon dioxide and water, you always kind of assume these hydrogens are those hydrogens. They're actually not. They're totally different hydrogens. Though those C's in the carbon dioxide are those C's from the glucose. Yes, ma'am?
Two things. Gelling is very interesting.
What did I say?
Briggs complex 4.
I think that's my ego coming in. I'm just going to refer to anything that's science now as joeing.
And do you scroll back up to the links of this?
Yeah. So we're not going to look at how ATP synthetase works today. We're obviously going to do that on Monday. Monday, we're going to do that. Tuesday, we're going to do a lab. By Monday, I hope to give you every single thing you've given to me back and to give you your current grades. I don't put grades until I have a whole unit's worth of stuff put together. I've got some of it marked. I don't have all of it marked. I will have all of it marked by the end of this weekend, come hell or high water. Yeah. Oh, then we'll just shift everything. Like, are most of you going? Hands up if you're going to Brock on Monday. That would be helpful. Yeah, okay. Then just everything I said, shift today. That's fine. Sorry? I'll give you your first assignment for this unit Monday, and I'll post it so you guys who are away, you can also just come get the physical copy before you go. But listen, I cannot stress enough, the best way to understand this stuff, make sure you read that textbook. How many of you actually have? How many of you who actually have found it useful? I think that, like, learning how to read a textbook is so helpful. And if you don't find it useful, look at me, this is important. If you read the textbook and don't find it useful, and this isn't me criticizing you, but it's the truth, like, that's not the textbook's fault. The textbook is such a wonderful distillation of this. Textbooks are, they're so much better than the internet. Like, Wikipedia is actually way too complex. What happens on Wikipedia every time you go to an article is that the people who study this for a living get their hands on it, and they put so much really high-level information in it that it's really hard for you to follow on a basic level most times. And then, like, other online sources, you can't necessarily trust. There's a lot of nonsense out there, especially now in the era of AI generative stuff. I used to feel pretty good about trusting online stuff as ways to learn. Now, more than ever, give yourself the process of see if you can trick ChatGPT into telling you that you are a world famous hockey player. And I promise you can. If you ask the right questions before you know it, ChatGPT will start telling you all about your biography and where you were born and all this stuff, and it'll all be nonsense. It'll all talk about how you're the best player on the Montreal Canadiens. You can get to say anything. And the problem is that it just makes stuff up. So now, when most places are laying off their writers because it costs money to hire a guy who studied science to write articles, boy, you know what's free? Having it just linked to a ChatGPT bot that just creates an article on the spot, but you can't trust it. But the textbook, and then YouTube videos might be made by somebody who knows what they're talking about. They also might just be made by some schmuck who thinks and people love to think they know about science. So get in the habit of learning how to read a textbook for information. It really is the best source available. I'm in the process of trying to buy them. I don't quite know what to do because buying one set of textbooks will eat up approximately 70% of my science budget for the year, and I don't quite know how I'm going to pay for gloves. So I'm working on it. I'm thinking of begging my parents for money for textbooks. It's a true story. Like, hey, remember in first year university when you lent me money for textbooks? Well, I'm here to ask you to do it again. By the way, I'm going to need 68. I think they will probably say no. I'm gonna try. Listen, let it never be said that I am afraid to beg my parents for anything. You know, they knew what they were signing up for when they had a kid. Or maybe they didn't, I don't know. Yes, ma'am, sorry.
So how's it pumping 3 H plus?
Because there's always H plus in water. Did you take 4U chem already? Yeah. So you know that, because water just left to its own devices, what does water do? All water exists in equilibrium, right? Now, the truth is we think of it as being H3O plus and OH minus, but for the purpose of this, let's just say H plus and OH minus. So we always have this available. Anytime you have water, you have H plus to work with. And you have OH minus to work with, but in this case, it's the H plus that cells use. Did that answer your question?